So here are our C3 and C4 plants. This is our C4 grass. Here's our C3 grass. And what I want to do is I'm going to take the chamber and we're going to basically put pieces right along the bottom of the chamber. So we want to cut them so that they're the right length. And then we're going to line the bottom of the chamber with these pieces. So here's my chamber. I'm going to place this in here. We're going to line the bottom of that nice and flat. We'll put another one nice and flat in there. Here's a third one nice and flat in there. Okay, so we can see all that. I'm going to cut a couple more. And then we'll be ready to do this experiment. So here we go. Here's another one, nice and flat inside, and a last one nice and flat inside. Okay, so now I'm going to move these out of the way. This is our C3 plant we'll do first. I will then go ahead and hook up our CO2 gas probe, and we will place on a heat sink, okay, on the top of that. All right. And so once we get that heat sink now on the top of it, we shall be ready to add the lights. And we'll basically get this light on top of it going. And this is a nice big broad spectrum light. And then that should then maximize our photosynthesis so that we can go over and take the readings on the Logger Pro program. Okay, so now I'm in Logger Pro. I want to set up an experiment. So I'm going to go up to experiment. I'm going down to data collection. I want this to be in minutes, not seconds. We're going to run this for, let's say, 15 minutes should be more than enough time. My sampling rate, I want a lot of samples per minute. So we'll go about 15 samples per minute. And I'm done. So here's my y-axis. I have CO2 on my y-axis. So I'm going to click on this 5,000. And since my value of CO2 down here is like 420, we're going to change that to something more appropriate, maybe like 700-ish, around there. Okay, now I'm all set and ready to go. Okay, so we're all set up with our C3 plant ready to go. So I'm going to go up here and hit play. And then we'll start our collection. You can see our live feed here, 4:35. It'll take a few minutes here for photosynthesis to fully kick in because I just put the lights on. But as soon as photosynthesis kicks in, then it's going to go down. We don't have to worry in this instance about dark versus light because respiration isn't a problem in this situation. So in this case, then we're just going to put it in the light. And the key thing we're looking for is when it goes down, where it equals out, what level of CO2 is going to be where photosynthesis equals the rate of respiration. So rather than you just continuing to watch this, I think I'm going to turn it off and then we'll record more at the end so you can see the end results. Okay, so now we've run this for a little bit over 10 minutes. Okay, I extended the time a teeny bit just to, because we didn't quite get there in 10 minutes. So we can see up here, we see this line, our photosynthesis is faster than respiration. It's going down. You can see this curve here. This curve is because photosynthesis is slowing down, while respiration is staying about the same amount in the background. It's coming in here, and you can see here, we have a teeny tiny, a little bit of a curve down. It's almost fully stopped on here. Over here to the left, then we can see our values. We can see that our lowest value is 222 parts per million. And we can see that that's that data point right there is 222. It's staying around 222 here. Went back up to 223, so it looks like it's leveling off right here. So here's our level off point. If we left this running, it would continue to go nice and straight, where due to photorespiration, photosynthesis slows down due to CO2 being the limiting factor, and then due to photosynthesis, then it speeds up. So we're going to go ahead and stop this. Well, now it went down to 217, then it went back up to 222. So we'll stop this. And so this is the value we want. So you want to look at this and find the lowest value associated with that. And we'll scroll down to the bottom again so that you can see the lowest value here. And you'll record the lowest value of CO2 where basically photosynthesis finally stops and equals out to respiration. Okay, now we're done with that trial. We'll move it back again, turn off the light, move these out of the way, take this out. We'll remove all of these green guys in here. Then we'll go back and grab the C4 plant and do pretty much the same thing, where for the C4 plant, I will take leaves and we'll get those about the right size. And then we're going to put them in nice and flat along the bottom of our tube here. So you have one nice and flat one. And the reason we do that is that we basically want to maximize the rate of photosynthesis. So we want as much light hitting as much surface area of the plant as we can. So if we maximize that light, then we can get that line to go down. Because this will go slow or fast, the higher or maximal photosynthesis we get, the faster this experiment goes. So we'll move this then, and we'll go back here. I'm actually going to air it out a little bit so that we return all of the air back to its original amount. So we're starting at the same amount. And we'll add this. Then we will put on our heat sink and get our light down right on top of it. Okay, now here we go, and we're ready to do our next round. Okay, so now I have my C3 plant, or my C4 plant all set up. So this will be the C4 plant. The first one run here was our C3. Now the second one run will be our C4. So we're all set up. It's been acclimating for a little bit of time, although in this case we don't have to worry about it because we want to see the overall trend. So I'm going to hit start. We will store our latest run so we can see both curves, and we are all set and ready to go. So there are our values. Okay, so here we are at the end. We can see that this is our C4 curve, and we can see that the photosynthesis was going really well through here. Don't necessarily compare the photosynthesis lines because that depends upon how the light was hitting the plants and so on and so forth. The key thing we want to look at is where this line levels off. So this line levels off here, and this line levels off here. So over here, if we look at our values, I'll go ahead and stop that one. We'll stop the collection because it's looking good. And then we want you to record the value where it leveled off at the bottom. So let me scroll down here. And we can see here, you want the lowest value of CO2 where we get that equilibration point, okay, where photosynthesis equals respiration.